So today, Sergio and I are going to um, walk you through, um, really the central part of single cell analysis, which is identifying the cell subsets. Um, so first, let's just start by, remembering what, cell types are and what they look like. So to motivate what we're doing. So here we're looking at microscopy images of different healthy tissues throughout the human body. So we have the skin, your skin layer, part of the brain, blood vessels where you can see different immune cells migrating into the tissue, the very nice sort of patterns in the small intestine. And then on these last two images, we're seeing diseased tissue where there's this kind of change in the organization of the tissue. It's a lot more disorganized typically. And so, in all these examples, the cell types and states are going to be different. There's going to be tissue specificity. There's going to be things that are specific to the disease. So really, what single cell transcriptomics and what these analyses are going to show us are those differences and potentially what genes and gene programs explain those differences. So yesterday, Daniel walked you through the beginning parts of the typical single cell transcriptomic analysis. So we start with this pre-processing of the data, which is taking your raw sequencing data coming off of the sequencer. So you've done your experiment, you have the raw sequencing data, and you build an expression matrix that has the expression of each gene in every cell. And there's usually 20,000 genes roughly in a human. And, there could be hundreds to thousands to tens of thousands of cells, depending on which technology you used. Then Daniel talked about filtering the cells and different types of quality control you need to do on your data because, these experiments are pretty noisy. They're pretty difficult to do. So there always needs to be a step of assessing the quality of the data. And then finally, Daniel talked to you about normalization, which was for every single cell, normalizing the counts of the genes in the cell to sum to a constant number and then taking the log of those counts. And that was to correct for differences in sequencing depth across individual cells or across batches of cells. Yeah, so any questions on what Daniel had presented yesterday? Okay, well, if there are questions, just you can raise your hand or use the chat or even just start talking. Okay, so then what we're going to talk about now, Sergio and myself, are the clustering aspects and the biology aspects. So clustering, how do we actually organize this data into clusters? So those clusters are going to represent the different cell types. And the biology, how do we know one cell type is one cell type? So how do we know once we've clustered the cells that we're looking at T cells versus epithelial cells versus lung cells and so forth? Okay, so to start, our data exists in very high dimensional space. So 20,000 dimensional space, one dimension for every single gene. So normally we think in like 2 or 3 dimensions, like if we're following a map, like Google Maps, that's 2 dimensions. If we're, walking around, we're in 3 dimensions. So 20,000 dimensions is very hard for people to reason in because it's, just not, it's outside our normal experience. And, and the other problem with this, other than we have a hard time imagining it, is that mathematically, a lot of the things that we do tend to fall apart in very high dimensional space, and distances between objects stop making much sense. And we care about distances between the cells and how different they are, because that's going to be the basis of saying, how different or how similar they are. And that's really how we do clustering. So is there a way to kind of deal with this issue that the cells are in such high dimension It's kind of a great thing that we've measured all these things, but it's also mathematically very challenging. So there's a few things to know, both technically and biologically. First, technically, if you remember, that single cell data is very sparse. So most of our measurements are actually zeros, and the data is sparse because experimentally it's hard to capture every single transcript in a cell. That's one aspect. And then the other aspect is that not every cell expresses every gene. So, a T cell expresses one set of genes, and an epithelial cell expresses another set of genes, and a pneumocyte in the lung expresses another set of genes, and so forth. So, a lot of the times you don't need 20,000 genes to describe what a cell is doing. And also, technically, we haven't really really well measured all those genes regardless. So there's an argument that we can reduce the dimensionality because of these technical and biological reasons. And so how we do that often is we focus on those genes that are most variable across the data. And that just means that, the gene's expression might be high in these cells and low in these other cells. So we're just looking at variability. And variability is a really good signal because it means that that gene, might be kind of correlating with whatever the cell types that you have are. So in T cells, CD3 is a very important gene, and in epithelial cells, EpCAM is a very important gene. So those genes are high in those respective cell types, but they should be close to zero expressed in other cell types. So they'll have high variability. In contrast, a gene that's always at the same expression level across all your cells is interesting in a way, but it's not interesting in that it's going to explain the variability in the data, right? Because it's always the same. So we focus on identifying highly variable genes. And so here's a typical plot of how we do that. So these are all the 20,000 genes from a single cell experiment. And each of these dots is a gene. On the x-axis is the mean expression of that gene across all the cells. And on the y-axis is the coefficient of variation, which is it's the standard deviation divided by the mean. So what you can see is that there's this sort of background expression between the mean and the coefficient of variation. And then there are genes that lie off of that. And so the reason we're plotting things this way is because there's an inherent connection between, or relation between, the mean expression and the variance of the expression. So the more highly expressed a gene is, the more variable its expression will be. So you need to account for that underlying relationship. And so how we do that is by making these plots of the mean versus, this is one measure of variation. It's kind of normalized in a way, but the coefficient of variation. So there's this background that's represented by this sort of purple line. And then genes that are really far off of this background are more variable than we would expect by chance given their mean expression. And so that's how we identify like the most highly variable genes. So it's a little hard to see with these blues and blacks, but the genes that are the most variable have these blue circles. Okay, so that's where we get the variable genes from. And then at this point, we ignore all the other genes while we're doing our clustering analysis. We don't ignore them forever. We'll come back to them when we're thinking about the biology, but for clustering, we're going to just use the variable genes. Any questions on that? Okay. So the next step is another step forward in reducing the dimensionality even more. So as I said before, the cells are in 20,000 dimensional space. And the genes, some genes are lowly detected, and some are just not expressed because of the biology of the system. Another really fundamental aspect of biology is that the genes are not independent of one another. Why are the variable genes used? Okay, so Tina, so let me just, before I go into this, Tina has a question. Why are the variable genes used in clustering? So clustering by definition, it's for separating things out so similar things are next to one another and dissimilar things are far apart. So things that are always the same are not going to help you kind of compare similar things or dissimilar things. So if you have a gene that's always on, sometimes these are called housekeeping genes because they're just so crucial to the function of the cell that pretty much all cell types have them. Those genes are not going to be very useful in separating things out by difference. Versus variable genes where it's high in one cell type and low in another, that's going to help group cells that are similar and group cells that are dissimilar far apart from one another. So remember, clustering is all about finding similarities, but it's also about separating things that are dissimilar. So it's those two aspects, the kind of the yin and the yang. And yeah, so hopefully that answers it. And then, okay, Tuna got it, great. Okay, what are examples of non-variable genes? Oh, yeah. So non-variable genes are genes that are, like I'm saying, are these so-called like housekeeping genes is often what they're called. And they are genes that are involved in like fundamental cellular processes that have to happen across all cell types. So I think like actin, which makes up the cytoskeleton, is often one of them. There's like a list I can get for you. I just don't remember what's in that list. But there's a few for sure that are commonly used. It's also they're not excluded forever. So the clustering is just to identify the cell types. But once we've identified the cell types, we go back to looking at all of the genes. So this is just for the clustering. And the CV is so the CV is the coefficient of variation. And the mean counts are, so there's 20,000 genes here, each is a dot. So this one here, HBZ, what this is the mean expression of HBZ across all the cells. So if cell 1 has expression 5 and cell 2 has expression 10 and cell 3 has expression 15, then the mean is 10. So that's what that is. And then, okay, with the so-called, yes, and then Daniel's question, are the housekeeping genes filtered out before clustering? We don't really need to filter them out explicitly. Like we do have a list of them, and if you're really interested, I can share with you a list that I commonly use. But we don't need to filter them out. We just need to filter out things that are not variable. They often filter themselves out because they are often the genes that are close to this background, because they're just kind of like not very variable by definition because they're found in all the cell types. Okay, good questions. Thanks for asking all those questions, Daniel, Tina, and Luis, and feel free to ask more. Okay, so now we've reduced the dimensionality, and so when you do the variable gene selection, you often go to about 2,000 genes. You go from 20,000 genes to 2,000 genes. And then what we do is another step called kind of this further dimensionality reduction. And the basis of this one, which is going to be on principal component analysis, or PCA, the basis of this is that genes aren't independent of one another. So there's genetic regulation where a transcription factor will turn on multiple genes at the same time. So if you have a transcription factor and it's turning on genes A, B, and C, when you see in a cell that gene A is on, you have a pretty good guess that genes B. So knowing the expression of one tells a lot about the of the other two genes. They're in this, they're regulated together, they're in a regulon or transcriptional program or whatever name you want to give it. So they're not really independent. And that actually is really great because that means we can treat genes A, B, and C as almost this group that is either on or off together. So we don't need to think of three separate genes, we can think of groups of genes. So one method to identify those groups, so obviously it would take a lot of knowledge to know, genes A, B, and C are kind of working together. So what's like a quick way to kind of automatically identify this? So principal component analysis is one of those methods. And how it works is, let me just go to this slide right here. How it works is, imagine you have a sort of simple where you have cells that only express two genes, X and Y. And this is the expression cell 1 of X and Y, cell 2, cell 3, cell 4, and cell 5. And what you can see is that, pretty much that these lie on almost like the line Y equals X. So each of the genes X and Y are kind of like very related to one another. So as X increases, Y increases. So that's using two dimensions to describe the data. But if you're clever about it, you can really think that this data is in one dimension. Imagine if you fit a line through this data. Now everything is lying on a line, pretty much, rather than in this plane of the X and Y axis. And that's exactly what principal component analysis learns to do. It finds the direction of the maximal variation and, in a sense, fits a line through there. And that becomes your principal component 1. And this is a new component, a new kind of axis. And now it looks and says, okay, how much variation is left and how do I explain that? And I explain that remaining variation that's going to be orthogonal using principal component 2. Is there a question? I think there is. Okay. I'll make a note of that. Okay. So now your data is really just described by how far along you are in principal component 1. And that's just really like, imagine you're just fitting a line through here and you're looking at where cell 1, 2, 3, 4, 5 are along that line. That's principal component 1. And then you have or sort of uncorrelated with that, or you try to explain the remaining variation. So once you fit a line through here, basically most of these points lie pretty closely along that line. And so there's not really much more variation to explain, but that remaining variation is explained in principal component 2. So I'll show you another cartoon of this that I like because it takes a little bit of, if you've never seen this before, it's a little weird. Let me just move things around and zoom. So here's a cartoon of that. So here now we have, again, a plane, and we have points on the X and Y axis, cells. And we're trying to, do principal component analysis. And so we're shifting this principal components 1 and 2, they're kind of rotating around. And so what you'll see is when this long line here, this is principal component 1, when it lines up along the direction of maximal variation, these little distances in red between the points in the line are minimized. So you just saw that right there. So just look again, and what you're going to see is that, when that long line lines up in the direction of maximal variation, which is basically this way where my mouse is, you see that the distance between these blue dots and the line is really minimized, and they're really lying very far along that line. Now, orthogonal to that is principal component 2, which is going to try to explain the remaining variation once you've projected these points onto this sort of principal component 1. And that's where they end up. They're going to be somewhere along principal component 2. So qualitatively, what you can remember is that the first principal component is going to explain the most variance in your data. And variance in your data just means it's spread out. So here you are spread out along principal component 1 once it aligns right here. So the points are spread out. And then the second principal component, which is going to be uncorrelated with the first one, it will contain as much variance as is still remaining that it hasn't been explained by the first principal component. And then you can keep doing this in higher dimensions. So here I'm just showing you examples in 2 dimensions, but it really can go into 3 dimensions and 4 dimensions and so forth. How many principal components are appropriate? So that's something we'll look at in the lab. Good question, Luis. It's something we'll look at in the lab. Generally, it's somewhere between 10 to 100 principal components, but there's heuristics to ask, when we're reducing the dimensionality of the data, how many principal components do we need? Yeah. And let me explain it. I'll explain it one more time, just for Karut and anybody else. So going back to the probably simpler view of this, we have data in 2 dimensions. And the point of principal component analysis is to reduce the dimensionality of the data. So right now it's in 2 dimensions. So the only way we can reduce the dimensionality is maybe going to 1 dimension. And how we do that is you can think that there's a line that's going to be fit that goes through the data and gives you sort of this best fit. and the best fit line is going to explain the most variation in the data, and it's going to also minimize, how far apart these points are from that line you fit. But you're going to fit a line, and you can think of it as, let me see if I can annotate here. That's not very good. Move it. Okay, so you're going to fit a line through it, and that's your principal component 1. And then now you can think, okay, in this view, we're looking at where you are along principal component 1. So some cells are to the left, some are to the right, and that's what we're seeing here, where cells are to the left and further to the right. And then the last part is principal component 2, which is really like, it's orthogonal, 90-degree angle. And for that, we're just seeing how far is every cell from that principal component 1. So in reality, there's not much distance that these things are along principal component 2. So it's pretty flat. Those values are close to 0. Okay. Great. And there's a lot of resources that are online that kind of explain this and show you pictures, and so keep looking at those if this is still strange or confusing. But you're going to see this a lot in genomics. Okay. So then the last step really is, now that we've done the principal components analysis, and we have gone to a lower dimension, and as Luis was asking, how many principal components are appropriate, it's usually going to be between 10 to 100. So in this example, we went from 2 dimensions to 1 dimension. And in our single cell data, we are going from 2,000 dimensions, which is usually the number of variable genes we identify. We're going from 2,000 dimensions to somewhere between 10 to 100. And now our dimensions are combinations of genes or groups of genes rather than individual genes. So, principal component 1 here is a combination of the measurements of X and Y. And same with the gene expression. It's like, principal component 1 will be some sort of combination of all your genes, where some genes will be very important and some genes won't be very important. Okay. So now we're going to do clustering, and clustering starts by getting distances between pairs of cells. So the cells are now described by their principal component scores, and we're going to get distances between those pairs of cells. So it's just like doing things on a map in 2 dimensions, except now we're in maybe 10 to 100 dimensions. So we're just getting Euclidean distances on this sort of between the pairs of cells, and then we're going to make a graph and then cluster in that graph. So to start with, I'll remind you that there's many ways to cluster data. So these are just different data sets of points in X and Y. And their true identities here, what cluster they belong to, are kind of colored in orange, green, and blue. And that's the distribution of these points in space. And then on these columns are different clustering algorithms. And you'll see that depending on what algorithm you run, they will assign different cluster identities. So in this case, we don't really know like what's the ground truth in this sort of cartoon, like what's the real cluster identity. But let's just start with like a really simple case, like this one at the bottom here. Like it's really clear how to cluster this data intuitively. Like we group together points that are close together and points that are far apart, those become separate clusters. But when we look at this data here, like these like kind of dashes of cells, if you will, like some of the algorithms kind of give every one of these dashes a different color. Some of them kind of group things in a way that's a little unexpected. And the same thing, if you look at these circles here, like kind of intuitively, you would think this inner circle and these outer circle are separate clusters. But if you do something like k-means clustering, you'll get something a little different. So the point is that depending on the clustering algorithm you choose, you will get different results. So that's something very important to keep in mind. So let's go through quickly how most people do clustering, or one of the more popular algorithms. So you start with your distances between pairs of cells. And then what you're going to do is take every single cell and ask, who are my nearest neighbors? So for instance, this cell, we've gotten all the distances. So some cells are close to this cell and some cells are really far apart. So we start with asking, who are my nearest neighbors? Who are the cells that are closest to me in space? And so this is the nearest cell, number 1, and then 2, and then 3, 4, and 5. And then we do the same thing over here. For every single cell, we ask, who are my nearest neighbors? So I'll show you that again. This is nearest neighbors of this cell. That's an example for that cell. Now for this cell over here, we do the same thing. Here's its 4 nearest neighbors. And we can keep doing that. And so that's where the little connections come from in this graph. And you can see that, once we've built the graph, it already becomes kind of visually apparent, where the clusters are. And this is called K nearest neighbors, a K nearest neighbors graph. And K is the number of nearest neighbors we tend to look at. And then finally, the last step of the graph is what we do is we look for shared nearest neighbors. So let's take these 2 cells here and here, this sort of green and cyan cell, and we ask, here are the neighbors of the green cell and here are the neighbors of the cyan cell. And you see that they actually overlap where 2 of their neighbors are the same. So this one has 4 neighbors and this one has 4 neighbors and 2 of them overlap. So that's called having shared nearest neighbors. And that's the ultimate basis of how we put edges in the graph. So 2 cells are connected if any of their nearest neighbors are shared. Okay. And so now, once we've built this sort of graph and things are connected by their sort of shared nearest neighbors, we can start to do clustering on that graph. And the reason we've gone from distances to a graph where these edges now are a little kind of different is this lets us work with different levels of distances. So sometimes you'll do your principal component analysis and these numbers here will be very small, these distances, and sometimes they'll be very large. But when you build these graphs, you lose the dependency of that sensitivity to very small distances or very large distances because you're doing things just based on who's closest to you. It's not, are you 1 unit away versus 2 units away versus 3 units away? It's like literally who's closest to me. So the actual absolute distances start to matter less. Okay. So what we're going to do is we've made this kind of graph and we're going to do clustering on the graph using a method called community detection. There's many methods to do community detection, Louvain, Infomap, Walktrap, and you're trying to optimize something called the modularity. But what you can think of the modularity as is just, again, grouping together similar things and keeping dissimilar things far apart. So for instance, if you had a graph that looks like this is this identity where you have green here and purple here, this maximizes the modularity versus this identity here where you have 7 or 8 cluster identities. Each of these is a different cluster identity. This is like a really bad sort of clustering. Likewise, if you just color everything the same, that's also not so great because you lose the fact that this group and this group are dissimilar from one another. So this is the best kind of identity assignment for the clusters, a green cluster and a purple cluster. But, there's a lot of fiddling and kind of parameters you can change that will change the outcomes of these clusterings. And if you use a different algorithm, you might get a different result. So that's clustering. So that's another kind of set of complicated steps, but hopefully pretty reasonable and intuitive in some ways. Any questions on that? And just to remind you once more, the clustering is happening after you've done the principal components analysis. So the cells here are now described by principal components, and we're looking at how far apart cells are in that principal component space. Okay. Okay. So the last thing I'll talk about before handing it over to Sergio is identifying differentially expressed genes. So once we've gotten the clusters, we want to know what genes are high in one cluster and low in another cluster. So for instance, if you had a cluster of T cells, you might want to know what genes are high in the T cells relative to all the other cells. And more realistically, you won't know what the cell clusters are and what they represent until you start doing something like looking for differentially expressed genes or some of the methods Sergio will talk about. So in bulk sequencing data and bulk RNA sequencing data, you often have a gene expression that looks very nicely distributed. So this might be cluster A, and this might be cluster C, and you can just do a nice statistical test to say whether the means of these distributions are significantly different from one another. Now, in single cell data, as you remember, as you recall, a lot of our measurements are actually zeros. So we'll have distributions that don't look like these nice sort of Gaussians per se. Instead, we'll have distributions that look like they might have a peak at 0 and then another peak somewhere else. And so comparing these distributions is much more tricky, right? Because the mean, if you just want to compare the mean, it starts to lose a little bit of the meaning, no pun intended, because you have all these zeros here. So the more zeros you have, the more the mean is going to be shifted to 0. But a lot of these zeros might be because of technical reasons and not because actually your gene is really lowly expressed. So a lot of methods are kind of these like very nice statistical models that try to account for this effect. What we're going to do today are really, you're going to see some really simple methods that, just run out of the box, like Wilcoxon and t-test, which are kind of, well, Wilcox is like a non-parametric test, and t-test makes a lot of strong assumptions, but there are sort of probabilistic models, like based on the Poisson distribution or the negative binomial, that you can also perform to look at differences in distributions. And so this is just going back to the world of statistics now. This is not genomics or machine learning. This is just statistics you learned in your statistics class about how to say whether two distributions are different from one another. So there's lots of different ways to do that. People have written review articles where these are all different statistical tests for differential expression, and these are different metrics of performance on these sort of standardized tests. And so you can get recommendations for like what are good methods to perform differential expression, or what are good methods to try. So that article is down here. There are the reference to it. But the point is, like in anything in single cell analysis, there's often more than one way to perform the task, and some ways are going to do better and some are going to do worse. And this is pretty important that we identify differential expressed genes, not just because we want to identify cell types and cell states and so forth, but often, hopefully, we have some sort of biological question, like a case control type study, where we have a disease sample and a healthy sample, and we want to identify which genes are differentially expressed. And we don't want false positives, we don't want false negatives, we want to really hone in on the real biology. So careful statistical thinking is important. This is almost there at the end, almost handing over Sergio. And then this is like an example of what you might get out of such an analysis. Here are the cell subsets, like 0, 1, 2, 3, 4, 5, 6, 7. And here are the differentially expressed genes for each cell subset. And then the cells here are colored by the expression of the gene, where yellow is high and purple is low. And you see this block structure along the diagonal where these are differentially expressed genes, and they're high in cluster 0, and these are high in cluster 1 and cluster 2 and cluster 3 and so forth. Okay. And then finally, okay, so we've done the differential expressed genes. And the last thing I'll say before handing it over to Sergio, the very last thing I'll say is that often, or almost always, we come up with visualizations for the cells in lower dimensions. So remember, the cells are in 20,000 dimensions. And so we need a way to display them in 2 dimensions that's sort of interpretable to humans. And so to do this, we use non-linear dimensional reduction techniques. And what these techniques will do is they'll take your high dimensional data and they will put it into 2 dimensions. And the goal of the technique is that cells that were close together in the high dimensional space are also close together in the low dimensional space. So it's really usually trying to maintain local similarity that was in high dimension, maintain it in the lower dimensions. And so here are just examples of dimensionality reduction techniques. This is one that's called the T-SNE. And so imagine these are two interlocking rings here in 3 dimensions. And now trying to show that in 2 dimensions, one depiction could be this, which seems pretty, sort of reasonable. Another depiction could be this, where they're sort of interlocking, and these circles could be closer or further apart. Usually the global distances, like 2 points that are far apart in this high dimensional space, usually those distances not preserved in these lower dimensional representations. Here we have something, oh, I forget the name of this. I think this is like a trefoil or something, but this is, this weird interlocking thing in 3 dimensions. If you were to try to project it to 2 dimensions, it's a circle. That's actually pretty accurate, but this might also be another way to show it, these other forms. So the most popular ways to do these visualizations are TS&E and UMAP. And you can go to, I think if I opened it, oh, that's my email. If you go to this website that you'll have the link to, you can kind of play around with making these lower dimensional representations. And this will give you a lot of intuition, this website, as to how these things work. So I'll put it in the chat. And with that, I'm going to hand things over to Sergio. Oh, actually, I'll take a second to ask, just to recap things, what we just did. So we talked about, so far, we've talked about quality control and normalization with Daniel. And then I just talked to you about variable gene selection, dimensionality reduction, making these distance matrices, and doing clustering, and then differential expression, and in these lower dimensional visualizations. So are there any questions before I hand it over to Sergio? Why unsupervised clustering? So we're trying to learn representations in the data that we don't already know beforehand. So we don't know that there's going to be T cells or B cells, or we don't have prior knowledge about the structure of the data. So that's why it's called unsupervised clustering. We really just want to discover it without telling the algorithm beforehand what to look for. However, once you've done this once or 2 times, say you've done this for your system of choice, like you're studying heart tissue, after that, when you get another sample, you could actually do a form of supervised clustering where you take your new data and project it onto the old data that already has the annotation. So that would be a form of kind of supervised clustering. Good question, Luis. But usually we don't know beforehand, what's the structure in our data, so we don't have anything to supervise. Any other questions before we go to Sergio? And I'll stop sharing so he can share. Okay, well, feel free to, keep putting things in the Slack channel.
I think you might be looking. Okay, I think you should be looking at my presentation screen, right?
Yeah, I see it, Sergio. Thanks.
Awesome, thank you. Hi, Orr. So, thanks, Orr, for the presentation. So I'm gonna continue the presentation talking about more like biological interpretation of the clusters that we already have. So once you've done all the pre-processing, all the clustering, the next point is to figure out what we have there, what biological populations do we have there. And I guess before doing that, we need to know what are we actually annotating. So there's another question. Is there any certain tools that can do the high to low dimension? Can you just go ahead with it without doing the principal components? Or do you want to answer that before I continue?
Oh, sure. Yeah. I can do the high to low. Can you just go ahead? I don't know of basically going to, there are people who will skip any kind of dimensionality reduction. They'll use all the data to do clustering. I don't think that works well typically. I think what we're showing you here is what 98% of people would do. So I would stick to going to the lower dimensional space. And let me just reiterate again that mathematically, a lot of these tools like clustering and and so forth just do not perform well in very high dimensional space. And there's even more modern techniques that we're not going to talk about here, like based on deep learning. That's a very popular buzzword right now, but those techniques also are based around going to lower dimensional spaces. So even developments in the field are still based around that central idea. So there's no, Tina, I would just kind of always use something like PCA or something fancier, but yeah, it's pretty important.
Yeah, and if I can add, I mean, it's also computationally a lot more expensive to not use feedback tunnel. Like if you try to do a new map in all your genes, it will take days. Well, if you do it in the PCA, it takes a couple of minutes. So it's computationally can also be very, very expensive. Unless you're working with certain types of single cell omics like CyTOF, where you have like maybe 30, 40 markers, then it might be feasible. But even then, like you might be losing some information. Okay, then if there's some more questions about the previous section. So yeah, so what are we actually annotating? So something to be aware is that cells exist along a multiple continuum. So something like what Orr said in the introduction, we have multiple cell types, but they're usually in a spectrum of stages. So we can have differentiation if we're working with a developmental process. We can have inflammatory response. If we are in a perturbation state, we have antiviral response or antipathogen response. If we are in an infection state, or we have other type of transitions like epithelial to mesenchymal transition in certain tissues. Which this means is that one specific cell type can be transitioned into another one. So that's something important to take into account because we necessarily will have always these great specific groups as a cluster, but a lot of the times we can have a differentiation continuum. So normally most of the techniques, as we have been discussing so far, put things into buckets or into groups. So we will have most of our cell types into a defined group. But this can be a bit arbitrary or not necessarily as specific. So this is something to take into account. And kind of having these unbiased approaches allows to appreciate how different cell types and states relate to each of across the spectrum. So yeah, so basically we kind of have to be careful when you define what a cell state is. You always keep in mind that you're trying to force a specific cell population into a data that is a bit less nuanced than that. So just to keep in mind. So what is actually notating cells in a single cell expression data? And what is the challenge? So gene expression is not discrete, but mostly a continuum, as I said before. And the other thing is gene expression doesn't necessarily mean cellular function. And a lot of and this is important to keep in mind is because a lot of the knowledge that we have for biological insights come from functional data, phenotypical data, that necessarily will correlate directly with gene expression. Meaning a lot of data that we have from like T cells or skin samples or any other kind of tissue that you can think of are in the protein level or the phenotypical level, which a lot of the time will match the expression of certain genes, but not always. So that's something to keep in mind. They're associated, but not necessarily the same. The other thing to keep in mind is the resolution. So for example, so depending the clustering that you do, you may have to have major cell types, less subtypes or subpopulations. For example, in this example that you see here, we could have 5 major cell types where we have like CD4 T cells, but we can actually go deeper and have subtypes of those CD4 T cells, and even deeper of these subtypes to go into specific stages of these cell types. And this will depend on how much dimension resolution we have, but also how many markers we use, what kind of markers we can detect. So there's kind of a balance into like what resolution we have and how much we want to discover. A lot of these subtypes will be small populations, will be really little amount of cells. So if you don't have enough cells, you likely will not be able to detect them. But at the same time, it will be difficult to like annotate all of this without the complete knowledge of biological information. The other thing to think into account is the phenotype. So the same cell type could be in different phenotypes. For example, as you see in this example, the same CD4 T cells can be different type of T cells, but at the same time, they can be in different stages. They can be inflammatory, they can be super pathogenic, they can be in different stages of the same backbone of cell type. And the other thing to think of is differentiation. So if you're working specifically in developmental processes, you will have transitions between different cell stages. And that will mean that a lot of times you will have intermediate stages as well, and things that can look like multiple cell types. So here I'm gonna talk to you about three main approaches to assigning clusters. The most simple one, and the one that is actually like the one that you can always rely on, is based on non-marker genes. So if your system is well known, you can use marker genes to annotate these. So basically previous knowledge, canonical marker information from like textbooks. The next one is using differential expression of the clusters. So basically comparing each cluster against each other and trying to find out what makes this unique, and then trying to compare this to literature. And the next one is trying to annotate using previously reported information, modern scores or atlases. There's a couple of things to take into account that we are not going to discuss that in detail, but is that even though you may have specific cell types, you will have a specific cases like doublets, which Daniel explained yesterday, which makes these things a bit more complicated in the sense that doublets are very weird. So a lot of the time you might think that these are real cell types, but actually it's just an artifact of having multiple cell types together. So this is something you really need to be careful on, because this could affect how you annotate and interpret your data. Low quality cells are also very important, mostly because if your cells were dying or if they were not very well detected by your single cell technique, they will look funky. They will look different. They will be expressing a lot of genes that will not make sense. So you have to be very careful that you are not like interpreting this as something that is more meaningful than it is. And the other thing is host pathogen annotations. So this is important because it comes into the round that you will not only have the cell type, but you will have a response to the pathogen, and at the same time you will be able to detect the pathogen as well in a lot of these cases, which will mean that you will also have like information of like another organism and another stages of infection. We will go a little bit more in depth into it, but just something to keep in mind. And then just to complement with what Orr was presenting about the clustering, there's some issues with clustering is that you could over cluster, and it's trying to subdivide your population into many more populations that are actually there, which will mean that it will be more difficult to define what they are. And then when you start comparing these against others, there will not be that much difference. There's the opposite side where it's under clustering, is that you are not dividing your populations into as many as you need, and then you will have mixed populations in the same groups. And then there's different ways to approach this, and it's iterative clustering. There's automated versions of this and more simple version, and it's basically trying to find this balance into like how many populations do you actually have there. And I think that in a lot of the cases, it's like something you kind of have to test yourself. It also depends on your question. If you're trying to evaluate things that are more like in a general space, you may just need major cell types, but you have very, very specific questions about a specific cell type. You would like to go as deep as possible and try to find as many subtypes and phenotypes as you can. So I guess it will always depend on what is your actual question, or what are you actually trying to solve. So the first type of annotation that we will discuss is annotating based on known marker genes. So basically, this just requires understanding of the expected cell types and their markers. So basically here you will have certain biological expertise of the model that you're working on. So if you're working with intestines, you should go back to the basics and trying to understand what is the intestine. If you're working with blood, the same thing. So here, many protein RNA markers overlap. So if you have information of like antibody stainings or protein expressions or knowledge from like protein atlases, they do overlap with RNAs most of the time, but not always. So you will have that a lot of markers that are well-known protein or antibody markers for certain cell types will not appear into your RNA space. And this is very common given that they can be post-translational modifications, post-transcriptional modifications, as well as different levels and amounts of what like phenotypically the protein may imply and the RNA may imply. So this is kind of like a miss sometimes, so it's just something to take into account. Even if you know your biology very well, that doesn't mean that you will always be able to find these markers that were traditionally used to determine what a cell type was. Yeah, so the same thing is like experience with cell types in sample and errors will help identify which protein markers do or not translate into the RNA space. So basically, you kind of have to try, test, evaluate, and then take it from there. In my experience, there's a lot of antibodies that sometimes were like knowledge that you know from decades of antibodies or like even textbook that will not be available at single cell RNA level. Not only because It's just not present, but sometimes we just cannot detect it. So this technique is really good, but it's also new knowledge. So a lot of these things will be new discoveries or new information that will not necessarily completely overlap with what is known already. So it is very useful to have these markers for like identifying cell states and cell types, but it's not necessarily that useful for annotating pathological cells in disease. Keep in mind that a lot of like during either physiological diseases like cancer or infectious diseases, cell states are an aberration of a stage. So you will have different things through like the normal stage, which not necessarily will express things that are well known or that necessarily make sense. So you kind of have to keep an open mind when you're working with like disease, because you will have a lot of things that are just aberrations of a normal state. So the easiest way to start evaluating differential gene expression is basically you compare one group against everything else, and you try to see what's upregulated or downregulated. Basically, yeah, you try to see what makes that group unique. There's different ways to do this. You can do this with differential expression, like a t-test, or there's more complex ways to do this. So no marking genes that are usually highly expressed will show up here, but this will also depend on the model. And a lot of the times, things that are very well known for a model, those are the top ones that are differentially expressed. So if you have very good markers, they may be significantly differentially expressed, but that doesn't mean that top differentially expressed genes. So just what makes the group more unique, that doesn't mean it's the things that are more known in literature about this. And when you're trying to do this, you can try to take the top 20 or 30 genes, kind of give you an indication of what is the cell state of the sample. A lot of the time, a specific phenotype will be the most things, the things that are more upregulated in that group, meaning like if the cells are in a stress stage or they're infected or they're doing inflammation or something that is transitional, this will be actively being expressed in that moment. It's an iterative process, so you should not expect to be able to completely identify all your cell clusters in the first pass. This will take some time, so you will have to like play into like, oh, this seems to be a mixed population, let's split it. Oh, maybe I have too many clusters, let's mix things together. So it's kind of something you have to evaluate and manually do a lot of the times, and it takes some patience. Likely there will be clusters that are very similar to each other. So when you run differential expression between those image clusters, and there's not really that many differences, it's normal to you mix them together again. Mostly it's because if you're comparing the same thing against each other, the difference might be just noise or things that are not necessarily that biologically relevant for the things you're studying. So basically, yes, you need to kind of continue iterating and recheck, at least to define what your populations are. So supplementary tables on paper is actually very useful because they will tell you different gene expressions that are normally used to assign clusters to a data set. So try to always go into papers and try to see what different people have reported of what a cell type is. This is also important, make sure species and systems are correct, especially if you're working with non-model organisms. Not necessarily the same genes that are in a species will be conserved across another species in the same cell type. So try to always be careful with this assumption. Even with simple things like comparing human and mouse, you will have genes that are not conserved across species. Also, technology use may have some certain influences. The detection rate of different technologies are not the same. So you might be able to detect certain genes in one technology and not with others. This in general should not be like really used in a vacuum. You kind of have to like really be aware of your context and kind of try to evaluate every time if what you're doing makes sense or not. So it's always useful to have a second pair of eyes. In my experience, most of the time is because you may be the first person annotating a cell type in some cases. And you may as well be not the first one and just maybe have done with thousands of times. So it's always good to compare to what is known. And if it's known, trying to like be sure and try to validate what you're finding. A bit more of an overview of host pathogens annotations before we continue into more specific details on how to do this is that in most of the single-cell RNA-seq techniques, you can capture anything with a poly-A. And a lot of pathogens, specific parasitic ones, and some like parasites, fungi, and some viruses have this poly-A. So in theory, you can also capture these the reads from these pathogens, which will allow you to, if you align it against the genome of the pathogen, also detect the stage of the infection stage of the virus or the parasite. And you can kind of have like a double readout, the host transcriptome, but as well the pathogen transcriptome. Something to only keep in mind is that detecting pathogen RNA doesn't mean intracellular pathogen. Mostly it's because you can have ambient RNA from the pathogen. You can have a lot of contamination. So it is not a perfect system. So if you are working with infectious diseases and in your cell, you detect certain levels of RNA from the pathogen, that doesn't mean that that cell was infected with this pathogen. It can also be that the neighboring were, and when this neighboring cells lies, you will be able to detect some of these. So it's something to keep in mind and be careful about it. Same thing, it doesn't mean there is an active infection, depending on the type of cell. For example, if you're working with macrophages or neutrophils, these phagocytes. So you may actually be able to detect pathogen RNA inside those cells, even though they're actually not being infected, they are actually phagocytes in the sample. And the reason why I work with this is because infectious disease, I mentioned this is because infectious diseases have become a very, very common use for single cell omics. For example, the COVID dataset that we're currently working on, SARS-CoV-2, it's a virus that is polyanated. So in here, we can actually detect the infection states of the sample. So this is, for example, an overview of the paper where this was published. And we can actually detect not only the cell types, as you see here, which will we evaluate a bit of this during the practical, but we can actually detect the stage of the sample. So for example, we use the metadata, we can see by PCR which the donors where the samples came from, which one were PCR positive for SARS-CoV-2. But then if we actually, and we can also associate other different type of metadata, like the level of the disease. But then we actually can also do is we can associate the amount of SARS-CoV-2 we detect per cell. So we can try to go back into the sample, look how much SARS-CoV-2 we detected per sample, and we can actually see which cells were infected, and out of each participant who had more RNA of the virus or less. And interesting, so like we can actually even do more things about it, and we can actually align the sample, the reads from the virus into the genome of the virus and try to see which parts of the virus are actually being more detected or not. Of course, depending the type, this was on the sequel, which is a 3' bias technology. So we detect more of the 3' of the virus, but we can actually see based on the on where this is mapping, what was an active infection or not. And the cool thing with this is that if you know the stage of the infection, we can actually start associating which cell types are more infected, which are more susceptible, which ones are responding to the virus, and they start not only looking into cell types or subpopulations, but actually active response and host pathogen interaction, which is something in theory you could actually evaluate with the data sets that we're working on right now. We will not go into detail into viral annotation or any of this, but it's just kind of like give you an overview of some of the power that this can be done and how you could annotate viral samples. We can also identify genes that are more present. If we cannot, sorry. So singular, the question in the chat is single-cell RNA-seq will pick up pathogens that are polyanated as well as the human RNA. So how could you distinguish between do transcript if you're looking for host RNA? So we will capture all of them. And when you sequence, we normally align into a genome. If you only align to the host genome, you likely will not, this transcript will just get filtered out and will not map. So normally what we do is that we create a hybrid genome that will be like the human genome plus the pathogen genome. And then when we map the genes, when we map the transcript, this will be mapped to the host RNA and to the pathogen genome. And then we can actually have a matrix where we have the genes from the host and the genes from the virus or the pathogen. So only for, again, this will only be for the polyadenylate virus, viruses or eukaryotic pathogens, but yes, we can actually differentiate those based on where they map in with the aligner, if they map to the host genome or to the pathogen genome. Yeah, so potentially you have pathogen genes, but you have to look for them. Yes, so if you don't use a pathogen genome, you will probably just miss these genes out because then you will not be aligned into anywhere. And this is very important because if you are working with samples where you don't know the pathogen, you might be missing this out. So you kind of have to have at least some a priori knowledge of what would you expect, unless you're using multiple like genomes. And then continuing with this, yeah, so if you are able to associate the viral amount to a cell, you can actually define what is an infected cell and what is a bystander cell, which actually can allow you to find different what is responding to the sample, to the pathogen, and what is not, as you can see here. So I guess that was the first approach, which is using marker genes, which in general is just basically trying to figure out differential expression between samples. But then a bit more in depth of that approach is, how do we use this? How do we use databases and literature to try to find out what are those marker genes, instead of just doing the differential expression and going back and looking what is conservated? We can do actually a bit more of like using what is already known in the literature. So there's like marker genes and databases that I previously mentioned. So for this, we are in general we can use different things, the literature, published databases, and single cell atlases. But there are also automated tools, which I briefly will describe, but very basically it's more a supervised way to do annotation and using things that are already different data, single cell data that are already published, and try to use that information to annotate our samples. So just going to the first one, marker genes and databases. So basically this is literature. It's very, it might sound very basic, but a lot of the cases, single cell RNA-seq might be new and a new field, but a lot of the cell types have been known for decades. And this knowledge is basically textbook knowledge. For things like immunology, hematopoiesis. Before I continue, there's another question. Will this be essential part of QCs publishing this kind of data? Yes, if you're working with pathogen sample, it is important to do this during the QC. You need to kind of identify what's infected, when it's infected from the beginning, because this will affect how you interpret the data. If you're comparing infected bystander cells and non-infected cells together, you will have a lot of noise and confounding factors. So if you're working with infectious diseases, this is something very important to take into account. So coming back to the presentation, so basically, for a lot of the samples, you should really go back to the book, to textbooks, to what is known already. Because for a lot of these things, like the signaling, the differentiation processes, what make a cell type unique has been known for years and years based on like standard biology and different approaches, genetic approaches. So a lot of this knowledge is already there. That said, not everything is there already. Like this is also how we do this, to discover new things, to find new biology, to find new insights. So it is important to use it, at least for things that are known, but in a lot of the cases, you kind of have to get creative and try to figure out what is there. And other suggestion, always rely on experts and collaborators. It's impossible to be an expert in every single field of biology. So a lot of the time, if you're working with a new tissue, you will have things that you might not be an expert. And it's always good to reach out to someone that might know already a lot of these things, so can direct you to where to find this information. It's a really huge amount of information that is out there, but it's also very difficult sometimes to figure out what is there. So regarding to tissue and protein databases, so there's already like a lot of information that is known from bulk, and this is still very useful. If you sort out a specific population, you can characterize it with bulk transcriptomics on protein transcriptomics on antibody stains, and this information is a good start to try to figure out what is there. There's a lot of databases that are very useful, in my opinion. We have the Protein Cell Atlas, which link is there, and you will have a go later on. There's the Immunological Genome Project. There is the Genotype Tissue Expression. All of these databases have bulk level and tissue level information, which will allow you to narrow your search for biological information. I think I have a video. Is this a video? Yes. So for example, so here I have a video of the Human Protein Atlas. Here you can put a gene, and this will actually tell you which tissues is the gene expressed. And if you actually go into it, they also have some single cell information as well. So you will be able to see what the gene does, where is it detected, if there's any association, and as I said before, which tissue expressed, but they actually will also tell you in which cell types sometimes have been detected. So it's already some information that you can use, as you see here, for example, which cell types can be, and it will allow you to have some information. If in your cluster, this gene is specifically differentially expressed, you can try to figure out with these databases why or where or what that can be. There's other databases for other organisms. There's a mouse atlas, there's the pie atlas, the CELLxGENE atlas, the zebrafish atlas, and as well as more smaller databases for other non-model organisms. So that's something to keep in mind. So for these main model organisms, there's a lot of information. If you are working with a non-model one, you can try to guide yourself by the closest related organism, but a lot of the time these things might not be super conservative. If you're working with major cell types, for example, if you're working with blood cells, this will be conserved across the tree of life quite well. But if you're working with specific cell types or specialized tissues, this might be a bit more difficult. And I think it's always very simple and although it's very simple and may sound very basic, it's always good to go to like the general databases like NCBI or the European Bioinformatics Institute websites where you can just put a gene and see which papers come out. And reading these things actually might give you a lot of insight or what something could be. And then regarding single cell atlases, there's a lot of, these were more like bulk atlases, but nowadays there's a lot of single cell atlases that come out as part of like the Human Cell Atlas initiative, as well as other initiatives that are out there. In a lot of these things, you can actually have a set of genes or your gene and search them one by one in these databases and trying to figure out what cell types have been reported that express this and try to associate your clusters of your groups based on this. So of course, we have the Human Cell Atlas catalog, which you can find these. We have the Tabula Muris, which is one of the biggest mouse atlas datasets. We have the Broad Institute Single Cell Portal, which is a compendium of hundreds of single cell studies where you can also do this. We have PanCloud database, which actually one of the favorites of mine, where actually this group compiled reported studies and try to find easy way to annotate cell types. They have the cell marker database, which is actually a compendium of cell markers. So for every cell type, we will tell you a list of reported cell markers. I will always say take all of this with a grain of salt, because a lot of the time your tissue and everything is context specific, so not everything that works for other people might work for you. So for example, for PanCloud database, let me just put this video. It's actually very simple to use. Here you can put, I mean, this one is focusing mouse and human, but you can put any gene in here. And if they have it reported, it would tell you things about this. For example, we have ApoA1, which is a marker for intestinal cells. And you can see here that it will tell you in which cell type it has been reported. You can see the study where these have been reported. You can try to go into it into a bit more of depth and try to see in the UMAP from that study, how is it expressed. And this is kind of a good way to like trying to figure out, hey, I have this. Does this cell type make sense? Where was it reported before? Oh, wait, this is a liver sample. I'm working with a liver sample, then it makes sense. This is a very patient process, and you kind of have to like go by this with a lot of patience and try to like be open-minded as well as you might discover. There's other ways around it. You actually can also look through cell types. So for example, here I'm going to see what are the markers that I can use for microglia, and it will tell me that these genes have been reported before in microglia and might be likely useful for you to annotate microglia if you're working with a brain sample. Okay. And then the automated tool. So this is a slightly different approach. So for this, basically, instead of going through the markers that we detect by comparing a cluster to differential expression, we're going to use cell atlases of previous studies to map this information in a supervised way into our sample. So basically, there are different approaches, but in general, this is kind of like a blast. If you are familiar with this, it's like mapping basically your cell types as a query into these databases. Something important for these automated approaches is that you need a reference. So you kind of need something that it's similar to your sample, that is previously been done, and ideally it's quite overlapping with what you have done. These methods have been very well optimized and in a lot of cases work interspecies. So you could use an atlas for a human to annotate mouse and vice versa, but it might not work perfectly. There's a question before. What are some limitations of testing using non-marker genes? So non-marker genes are very useful, but they are also, it depends where you're taking them from. If you are taking it from the same tissue, that might, from single cell studies, that may be very useful. But if you're taking it from a tissue that is not exactly the same, or from a different organism, or in a different disease, you kind of have to be careful with them because some of them might be conservated, but some of them might not. So they might not always work, or you actually might, it can also be that you may not be detecting it. So just because something is not being detected, that doesn't mean it's not there. So that's one of the main limitations, is that they are very limited. And a lot of the time you kind of have to like really like go gene by gene, marker by marker. And if you have something that hasn't been reported, it might make sense. You kind of have to just go through literature and see what it might mean. For example, there's a lot of things that you might only figure it out if you because they're part of specific signaling pathways, which might imply specific differentiation stages, which might not be necessarily what is being reported before. So going back to the automated tools, it can also done with different omics. So we're talking a lot of here about RNA-seq, but there are other methodologies like ATAC-seq or proteomics, where you could actually try to use the same kind of approaches. There are different ways to do this approach, and there's correlation-based and supervised classification. Correlation-based is basically you try to find which groups correlate the most with your sample, and then based on that, you will assign it. And supervised classification is using different machine learning approaches to annotate this. There's a lot of automated tools nowadays. This is just a small set of what is reported, but these work for like, they are both in R, in Python, in MATLAB. There's different approaches for this. So we have a lot of correlation based. Well, as I mentioned before, basically we use correlation to map either our cell type or our clusters to previously reported data set and try to see which one correlates the most. And if correlates high, then we assume that it's likely a similar cell type. And then we have the supervised classification, which again, there's a lot of them, and there's different approaches for this, which use neural networks, deep learning, autoencoders, random forest, different type of AI approaches to try to using the previously reported data set to infer populations in our samples. So correlation ones, there's multiple of those. Single cell map is one of those. And basically, as I mentioned here, you have like your query, which will be your sample. And then we correlate these against every single cluster in the reference data set. And then we try to basically map which one is the highest population, which one is the most similar population in the reference to ours. This can be done cluster by cluster or cell by cell, and it's basically done by similarities. And then we have the automated, the supervised one, where we try to annotate this in through different ways. So just as a summary of how I would suggest this should be done is you can always start with automatic annotation. Mostly because this will give you a global overview of what you might have there. Most of the time it's not perfect, and something to keep in mind. I'll give you a quick example in a little bit, but most of the time it gives you a rough idea what you will expect into your sample. But it always goes into like, you can start with the automatic annotation and you can try to improve it as much, but you will always come down to use marker genes, gene function, your clusters, and actually trying to define what you actually have there. Mostly is because, well, unless someone is doing exactly the same thing as you're doing in the same condition, your cell types are likely going to be different. And you're likely going to have different boundaries of the population on different stages. And then the last step that you always should do after automatic annotation and manual annotation is the verification, and which is actually trying to do experiments to validate what you're finding, trying to evaluate if your samples are present in other samples, consult with the experts, trying to see if you have the statistical support to say, hey, this population is what I think it is, to have enough cells to say, like, this is actually something. So a quick example, so this is a previous published data set where we were working with colon and ileum biopsies that were infected with SARS-CoV-2, and we did single-cell RNA-seq using the next genome. So basically, this is the kind of UMAP dimensionality reduction that we ended up with. As you may see here, we have multiple clusters. What we tried to do first was to do an automatic annotation of this, and using this paper that you see here. So these are organoids, and this was a published data set of human colon samples, so live tissue. So we try to use annotation from this data set to annotate our clusters. And how this will look like is like every single one of my clusters that you see here, it will be associated to a published data set from the previous paper, which actually gives me a good idea. So I can see like, oh, cluster 0 here is likely an enterocyte. Cluster 17 here, if I go back, is likely an enteroendocrine cell, and so on and so on. And this will give you a small idea of what I might have. The other thing that I can do is trying to do correlation. So here I have the published data set and my data set, and then I can try to see how do they correlate. Well, it seems like what I think is enteroendocrine cells correlate really highly with the published enteroendocrines, and so on and so on. This will give me an idea, and then the next thing I will do is actually look for the markers. So here I have like each of the cell types that I am notating and how is it expressed each of the genes. So I can see, for example, what I thought it was an enterocyte has a lot of these genes. What I think is a goblet cell have the marker genes present there. For example, this cluster, which previously was 17, which was mapped to enteroendocrine cells, I can map CELLxGB, which is one of the marker genes for enteroendocrine cells, and that way I can see actually, hey, it's super expressing that gene. So likely it is an enteroendocrine cell, so I can actually, with more confidence, say like, wait, it was automated annotated as that, my marker gene match, so likely it will be an enteroendocrine cell. And then actually, then I can also do like mapping multiple genes. So for example, the stem cells here, they should be high in LGR5 and SMOC2. So I can actually see that they are present, both of them are present there. So likely this population here, then are stem cells. And then after I have this cluster, I can basically then map this into cell types, and I can do like the differential I can actually, and with more confidence, say what each of them are. And if you actually do a heat map, you can actually see like the marker genes very nicely here in the diagonal, which is something that Orr presented before. And then if you remember at the beginning of my part, I mentioned that you really need to be careful with low quality cell types, because this might look like something that is biologically relevant, but it actually might be a technical issue. So for example, in this group, we have this section here that I here refer as low quality enterocyte. And the reason for this is that if I go and look at the number of counts that I'm detecting and the number of transcripts that I'm detecting, it's actually very low. And if I go into look what makes this difference, it's actually noise. So for example, in this one, I should have in the QC discarded these cells because they are actually not healthy cells. They were not well detected cells, but actually an artifact of the processing of the sample. And if I wasn't careful with this, I might be mistaking this as a real cell type. And this would be actually very problematic because I may be doing inferring biological insights for something that was just a technical problem. And yeah, I think that's everything from my example. If anyone has any questions, please feel free to put it in the chat or you unmute themselves.
All right, so I hope everyone was able to just stretch their legs, grab some coffee. Let's plan to resume now. So Orr and Sergio, I believe that we're going into a practical lab session now, so I'll hand the reins over to you.
Yeah, so hi everyone, welcome back. So now we're gonna go into the last part of the course. So congratulations, you're almost there. You know, and unfortunately, this is also maybe the hardest part. So let's all bear with it. But it's also gonna be good because it'll be challenging and hopefully you'll learn a lot. And you know, you'll have the code and you can always go back and ask us questions later. But this is definitely going to be you know, very interesting, but also a lot of our code you might not have seen before. And so please ask a lot of questions and, you know, don't get discouraged. This is always hard for people the first time they do it. Okay, so I'll share my screen. So you all should log into the RStudio server using the URL that you all have used the previous 2 times with Daniel's lab and Daniela's lab and right, and Sarah's lab. So reconnect. So log into the RStudio and then go and find the lab that's called, I don't know why it keeps doing this. Hopefully this isn't happening to you. HCA COVID-19 clustering lab.rmd. And you can find that by, you know, being in the files tab and clicking on that script. And then once you're in there, you know, you can see what the lab is about, but I will tell you verbally. We're going to be continuing to look at the COVID-19 single cell RNA sequencing data set collected from nasopharyngeal swabs from healthy and COVID-19 participants. Sergio just talked about that publication from his group, and Daniel and Daniela used that in the lab yesterday. I posted the PDF for the for that paper in the Slack channel, so you should all have it. And what we're going to do is we're going to try to recapitulate some of the analysis from the paper, in particular, finding highly variable genes, doing dimensionality reduction, clustering visualization, and doing differentially expressed gene analysis. So this should be fun. You know, if you get confused, like I said before, you can always go back to this. I also linked to a very helpful tutorial from the people who created Seurat that is based around looking at blood cells. So it's very, very similar in content. So that has all the images and plots all in one nice website. So it's easy to look at. Okay. And then the last wrinkle in this whole thing is that, we're all going to go into 4 breakout rooms, and each breakout room, in addition to running the main script, you'll also have a part of the script that you can focus on and try to solve those sort of questions there. And those parts of the script are labeled as group 1, group 2, group 3, and group 4. And so if you're in group 1, you just need to look at the group 1 section. And if you're in group 2, the group 2 section. So this is an addition to the main script. So we'll all be running the main script. So this chunk, and then this chunk, and then this chunk. But then when it gets to a heading that says group 1, 2, 3, or 4, depending on which group you're in, just do the one for your group. If you're really fast and motivated and everything's going well, you're more than welcome to do the other sections for all the other groups. But to start, run the main sections of the script, but also focus on, whatever group you are. And then at the end of that, what we'd like you to do is prepare two slides for your group that will have images or plots you generated, or an explanation of what you did or what you learned or questions you had, but just something to share with everybody tomorrow about what you learned. And that can, like I said, that can be plots, that can be an explanation of what you did. It could even be just questions you have or things you got stuck on, but just something that you will all create as a group together, and just 2 slides. And there's a question. Let me deal with that question later. Okay. Any questions about this before we get started? And while I have all of your attention, let me just remind you that you're going to want to change this home directory. So line 21. So I'm user SCW90, and you can find that really easily if you forgot your number. It's the same as before, but you can just go down. You're normally in the console, click terminal, and that number there, SCW90, is the one you want here. So if you were number 1, you just want to put a 1 there, and if you're 5, you put a 5, or 6 there, you put a 6 there. Okay, so pretty straightforward. Okay, I'll wait 10 seconds to see if anybody has questions before we go into the breakout rooms. Okay, and then with that, we're really fortunate that we have the CZI here with us, and the CZI is the Chan Zuckerberg. That they're mostly based in the Bay Area, and they are organizing a lot of the Human Cell Atlas efforts, and also spurring innovation by funding different areas that really need lots of people thinking about and contributing to. So, and they also are contributing a lot on the software level and in many different ways. And so they've been really partnered with the Human Cell Atlas for quite some time. And they are going to speak to us for the next remaining time about some of the cool tools that you can all use. And they're very accessible. So I think you can always reach out to them with questions, and they will be on the Slack, and they're happy to help. And yeah, we're very happy to have Pablo and Maximilian here to help. So I'll just hand it over to you guys and let us know if you have any technical issues.
Sounds good. Thank you so much, Orr. And thank you all for the invitation. We're very excited to talk to you about what we're doing at CCI, and also about this piece of software that we have been working on for the last few years that's called CELLxGENE. And I heard that you guys were doing some coding before, so rest assured, there's no coding in this session. That's the whole point of the software that we have been working on, so that anyone can analyze single cell data sets without coding experience. So hopefully this will be a smoother ride than the few ones that you probably had over the last 2 days. So I'm going to start sharing my screen. Please let me know if this is working. You guys can see slides.
Yeah, I see it in the Google Docs.
Awesome. So
Google.
now you can see the slideshow, that's right?
Yes, everything's good.
Perfect. Okay, as I said, we're coming from the Chan Zuckerberg initiative, specifically from the science initiative. There's also an education initiative, and we're going to be talking a little bit about what we do in the initiative, and then we're going to be talking about this piece of software called CELLxGENE. I'm Pablo Garcia. I'm a computational biologist, and we have Max here as well. He works on the product side, and he'll be doing part of the demo as well. This is what I just said. I'm going to go over what we do in the science overview, and then specifically about what we do in the single cell team, and then going into the bulk of the presentation, which is how to use this software called CELLxGENE. So our missions in the science initiative at CCI is to support the science and the technology that will make it possible to cure, prevent, or manage all diseases by the end of this century. We understand that this is a very ambitious goal, but we also put a lot of emphasis in the managing of all diseases, which means to be able to have a dignified life throughout disease. And we're putting a lot of effort into that area. However, we have more tangible goals over the next 10 years. And our main goal for the next 10 years is to advance the research and develop the technologies specifically to observe, measure, and analyze any biological process within the human body. So we're just measuring stuff inside the human body. And we do this by three main things. We fund, we do, and we build. So we fund, we give grants to scientific programs to advance fields. In the single cell team, we create grants for single cell research to occur or for analysis to be done on datasets that already exist in the single cell field. We do by working very closely with certain partners who perform the actual research and experiments to support them in any way we can, specifically on the tech solutions and the software solutions that they need. And we also build these pieces of software to help the entire community have access, equitable access to the data that's being generated through our grants. We measure our success in three different areas: by productivity, which means publications, preprints, softwares, grants. We try to have some reach, which means that we try to deposit all of the data and the software in public repositories. And we are very intentional about having open access, both in terms of our research as well as in terms of the software that we develop. And we collaborate with many folks in different areas. For example, this collaboration that we have with the HCA to provide workshops as to how to use our tools. So far, we have reached the 1,000 mark for the number of grants that we have given over the last 6 years that the Science Initiative has existed. A total of 58 different meetings and convenings where we try to bring people together to think really hard about problems and to provoke networking among the different grantees that we have. And we're very close to reach the billion mark in terms of dollars that we have given in the form of grants. Now, what we do in the single cell team. So talking about measuring biology in the single cell team, what we want to do is to understand the human body and diverse diseases at the level of the fundamental unit of life, which is the single cell. In other words, we want to be able to measure what's going on inside each individual cell of the human body. And we do that by accelerating the field as in terms of methods, technologies, and applications. So we have created a series of grants that are detailed here. I'm not going to go into much of the details because those grants are in the past and are getting to their conclusion. But essentially, these grants have been devoted to the generation of single cell data. And recently, we put out another grant that's called the Data Insights Network, which is devoted to the analysis and reuse of existing data. There has been 2 different cycles for this grant. The second one just closed, and the third one's going to open next year. So if you're doing any research that's related to data analysis, please stay in touch with us as we're going to reopen the last cycle of this grant, and we're going to be providing funding to perform this kind of research. And finally, we have developed CELLxGENE, which is a software tool that lets you analyze single cell data with almost no coding experience. So that's what I'm going to be talking about now. So this is the bulk of the presentation, and we're going to be demoing this tool. And please feel free to ask any questions in the middle of the demos as we're going to go through all the different aspects that you can use this tool for. So if you have any questions, either put them in the chat or somebody let me know that there's a question. We have two different flavors for CELLxGENE. One is called CELLxGENE Discover, and the other one's called CELLxGENE Annotate. CELLxGENE Discover, it's an online suite of tools and also a data repository that lets you discover, download, and analyze publicly available data. We also host data for free. So if any of you guys have any single cell data sets that wanted to be hosted online, we provide that service as well. And CELLxGENE Annotate, it's a tool that you install in your software. It's a small part of the portal, but you can install it in your desktop, in your laptop, and self-host it for sharing among your close circle of research. And Max is going to be giving a demo of that later on in the presentation. And I'm going to start talking about CELLxGENE Discover, which is the online tool. Before I go into the details of that, I want to give you a general overview of what happens with single cell data from raw reads all the way to process data, just so that I can put you in context as to why CELLxGENE is useful and on what steps along this processing of data CELLxGENE is used for. You probably, over the course of the last 2 days, you have heard a lot about all the different steps that are involved during the preparation of samples, isolating those samples, then sequencing the samples, mapping the reads back to the genome, and quantifying those reads in the form of matrices that are genes by cells, and then you have the counts for each gene in each cell. You have the read counts, and then you perform any further downstream analysis using these matrices. Well, CELLxGENE helps you specifically with the process data. So you probably learned a lot about what happens early on here, and maybe some of the coding exercises that you guys did also happen around this analysis area, where CELLxGENE is where it excels in grabbing these matrices and making sense out of them with specific tools that we have created for that. Okay, so I'm gonna get out of my presentation mode because I wanna share a link with all of you so that you can start playing around with the tool. So there's the link. Feel free to go ahead and click on it. There you go. So this is the landing page. As I said, CELLxGENE Discover, it's a standardized single cell gene expression repository. And you can start exploring the homepage. And as we go along the demo, I'll give you instructions as to where to go and how to follow the things that I'm about to show. Before I do that, I want to put a lot of emphasis on the kind of data that we have here in CELLxGENE. So we host these matrices that I just talked about, the gene by cell matrices, and we host the row counts as well as the normalized data. And then for all the cell metadata and gene metadata that we have, our data that we host has been through a very strict standardization process where we have put all of our data sets in the same vocabulary. So if you grab two different data sets, you can rest assured that the cell type labels are going to be equivalent among them and all the other metadata labels that I'm showing here. The same goes for gene names and gene IDs. This is actually one of the hardest aspects of being a computational biology and working with publicly available data, is that you always have to wrangle the data so that you can put it in the same space in terms of gene names, cell type labels, tissue labels. sex labels, age labels, so that you can actually make comparisons and correlations between all the data sets that you have. So we have made that heavy lift, and we provide all of the data in standard format so that you don't have to worry about that. There's three main things that you can do in the portal. You can browse data to find any data of interest for you. You can download that data in the two main different formats that are currently used in the single cell field. And then you can use some online tools to do basic explorations of that data. And I'll go into each of these different buckets and explain a little bit more as to what this means. So first of all, browsing the data. And here I'm gonna go into doing a live demo so that you guys can follow me. If my internet connection is good, then we can go ahead and do the live demo. If my internet connection is a little bit not good, then I have some animations prepared for you guys. But so let's give it a try. There you go. So let me just share one more link for you guys so that you can follow along. Right here. So if you go to this page, that's where I'm about to go. So here we have the collection page. We have two main ways to browse the data that we host in the portal. I'm going to focus currently in the collections way of browsing data. So here, if you go into the collections link that I just gave you in the Zoom chat, you can see all the different collections that we have in the data portal. If you start scrolling down, you can kind of see what they are. You have the title of the collection, whether there's any publications associated to it, and then tissue, disease, and organism that's coming from. A collection, it's a group of data sets that have some sort of relationship among them. Usually they all come from the same publication, or they are coming from a specific consortia that has given us their data. And here we have a series of filters that are pretty similar to like a shopping cart, where you can very easily find the data that you're interested in. For example, you may know of a specific publication, but you don't necessarily know what the name of the collection is. So I can look for specific authors and try to see if we have any data from those authors. And here we have 3 collections from Edlin. And you can also look for specific biological entities. So let's say you're interested in specific tissue or several tissues. So here we have made these filters very user-friendly so that you can browse the data very easily, where we have a three-tier system for looking for tissues. Maybe I'm interested in the nervous system, and within the nervous system, spinal cord or different parts of the brain, and I can go and look for what parts of the brain are available, and then start looking for the data that I'm the most interested in. For the following part of the demo, and we have filters for all of the labels that you can see here. A similar filter for cell types if you're interested in specific type of cells, development stage, disease. We actually have plenty of disease data. Most of our data comes from relatively healthy samples, but we have around in the order of 30 or 50 different diseases already hosted in the portal. And we only host data from mainly Homo sapiens and Mus musculus. We have some data from other apes, but that's only because we made an exception to host those data. But overall, you'll only be able to find human and mouse data. Okay, so I'm about to drop out another link for you. So this link that I just left there is for specific collection. So the collection that I'm going to be showing, it's from lung. So if I come to tissue, and then look for lung, and then it's going to show me all the different lung collections. And I believe the one that I'm going to be talking about is the lung map. Oh, this is actually the integrated human lung cell atlas. So here in a collection page, what you'll be able to see, it's a description of the data that's hosted here, and then a series of links that are associated to this collection. The publication, the contact that contributed this data to our portal, and then some data sources of other places where this data might be deposited. And then we have the ability to host any number of other links that you may be interested in looking at. And here we'll have all the different data sets that are contained within a given collection. And in this case, there's only one data set in this collection. Here, you can download the data, as I mentioned. So you can download the data in H5AD, which is for our Python users, or insert ROT format, which is for our users. I'm not sure if you guys have gone over the main toolkits that are used for single cell analysis, but these are the main formats that exist out there for the analysis of single cell data. And then you can either download it via the terminal or directly from the browser by clicking here on download. I'm not gonna start the download now because that's probably gonna eat up a lot of my internet. Okay. So that's in terms of browsing and learning more about the data, as well as downloading the data. Now I'm gonna speak more about the online tools that we have developed for you to be able to analyze all the data that we host in the portal. And again, I'm gonna share another link for you to follow along. And this is the part that I would really encourage to follow me as I demo, if you haven't already, because in my opinion, this is one of the most useful tools that we have hosted right now in our portal. So the link that I sent you is the link that takes you after you click here on Explore. So for any data set, you can click next. You can click on the icon that's next to the download button, and then it'll take you to what we call the Explorer. And the Explorer is a way to be able to analyze these data sets using an interactive tool where you can visualize first all the cells that exist in this data set in a two-dimensional canvas. Usually this two-dimensional canvas is constructed by UMAP, which lets you separate all the cells into defined clusters that are defined by their gene expression profiles. So it's just a way to nicely separate all the different cells in a given study and group them usually by cell types. So here in this tool, there's a few things that we can do. The most basic thing that you can do is start coloring all of these cells by the metadata that we have. So here we have the standard categories that I told you about earlier. These are the ones that we reassure you are going to be the same categories and the same labels across all the data sets that we have hosted in the portal. And then we have some author contributed categories. These are not standard categories, they're specific to each data set. So you can look at them and try to perform analysis if you know what you're looking at. If not, maybe not, you don't have to focus too much on this. And if I didn't mention this already, here on the left side, we have the cell categories, the cell metadata. So each cell, it's annotated to know what cell type it is, what depth stage it's coming from, what's the donor that gave this data, and a few other fields. So the first thing that you can do here is start exploring the data and coloring the dots using these different metadata. So here, if I click on this little droplet, what's going to happen is that I can see all the different cells in terms of the cell types that they're coming from. If I further click on this little arrow next to the category, then I'm going to be able to expand and see what are the different definitions for that category. So in this case, you can see all the different cell types that I have, and if I hover over any category, then they will pop up in the UMAP visualization. So that way you can keep track of what is each of these, where each of these different cell types is sitting on the UMAP. Another way to keep track of all the different labels here is for you to go and click up here on the three row icon, and that'll show you the categories, the label categories for the category that you are currently coloring by. So in this case, I just clicked on it, and it will essentially show all the different labels that I have here on the left side bar on top of each of the categories. So when we have a lot of cell types, as it is in this case, you'll see that it can get a little bit crowded, which is why we have it by default as off. But it's a good way to get an idea of the distribution of cell types that you have. We can also, using the scrolling functionality or using the magnifying glass here, you can zoom in and out of a cell in the UMAP, and you can click and drag to look at specific populations that you might be interested in. Similar to coloring, this what we call categorical metadata. So these are metadata that have a defined number of definitions. We can also color any continuous metadata that we have. For instance, for each cell, we know the number of genes, in this case, that a cell is expressing. So if I color by any category, by any continuous metadata, I will be able to see a color scale of what's going on with that specific metadata. So for instance, in this case, we can see that there's a few cells over here that are actually expressing a lot of genes. Whether this is a biological relevant piece of information or it's a technical artifact, it's unknown. And that's why you have to go and explore more of the data to see if that relates to anything biological or not. One way to do that is by coloring by any variable and then checking back other variables. So in this case, I'm coloring by the number of genes that any given cell is expressing. As I said, these cells over here seem to be expressing a lot of genes in comparison to the rest. And then what I can do, I can go back to look at my cell types. And then here, what you see now is that there's a little histogram that's appearing next to the label. So that histogram is based on the variable that I'm coloring on, so the number of genes expressed. And the more you see that histogram shifted to the right, the more genes that cell type is expressing. So here you can already tell, for instance, that acinar cells are one of those cell types that seem to be expressing a lot of genes. And we have some other cell types over here. And then you can start making sense of this data based on the biology that we know about these cell types. Okay. If there's any questions, please feel free to stop me at any point, because I'm going to transition to a different functionality of the tool. I'm going to go back and color by cell type, and I'm going to showcase another functionality of the tool, which is looking at individual genes. So on the left side, we have all the cell type stuff. And then on the right side, we have all the gene functionality. So here we can look up any gene of interest, select it, and then it will be loaded here. And once it's loaded, we can also color by gene expression, because this is actually a continuous variable. So here I can expand to see the distribution of gene expression across all of my cells, and then I can color by that gene expression. Let me zoom out a little bit. And similarly, as with what I just did, I can look at my cell types and see what are the specific cell types that are expressing that gene of interest. It just happens to be that I selected a marker gene of secreting cells, which is why we have that population on the right side of the UMAP being highlighted. And so you can explore any gene of interest and start looking at the distribution of the expression of that gene across all of your cell types. And finally, the last piece of information that I wanted to share with you is one of the most important aspects of this tool, which is performing differential gene expression using this, using the UMAP. So here, what I'm going to do, I'm going to pick the lasso tool. I'm going to grab, click, and drag. It might be hard to see, but what I'm doing right now is I'm creating a circle with the lasso tool, and then I'm dropping. And I just selected a few respiratory basal cells. And once I have selected them with the lasso tool, I can make them as my group one. So here you see that I selected more than 10,000 cells. And then I'm going to go back and select some of the alveolar macrophages. Same thing that I just did. And then click on my group 2. So I have 10,000 basal cells and around 4,000 alveolar cells. And now if I click on this little icon here, we're going to start performing differential gene expression. And this is something that if you were to do it with code, it could take a few lines of code trying to scratch your head around how to subset the data, get the gene expression data, and then perform any type of test. And now here, what we can see is that we have two gene sets. One that has the genes that are highly expressed in my population 1, which were basal cells, and then one that has expressed genes that in my population 2, which were the alveolar macrophages. And I can span these two lists to start looking at the different genes that are here. So currently it's loading because it's a list of around 15 genes, I believe. So if we let it load for a little bit more, you'll see first the distribution of the expression of all the genes, and then the distribution of individual gene expressions. And then I can just color by the gene expression of these guys. And you can see that the top result here is actually a good marker for the basal cells. And if I go down to see the other group of genes, then this doesn't seem to be that specific, but we can start exploring more. And there you go. We have the second top here that seems to be a very good marker gene for alveolar macrophages. Right. With that, I'm going to stop because I'm going a little bit over time based on where I want it to be right now. And I'm going to transition into a different tool. For that, I'm also going to share the link. Or if you're already in the page, feel free to click here on gene expression. And then it'll lead you to the latest feature that we launched in the portal. So this tool lets you explore gene expression of any gene of interest across all the data that we have hosted. So we have normalized and integrated all the data so that it's accessible to you without needing to do any coding. So the way this works is by you click in the tissue, you can add any tissue of interest. Let's say, for example, I'm interested in lung. Once you added the tissue, the second step is to add any genes. So I'm going to add the same gene that I have added earlier. And once we add any gene, what's going to happen is that we're going to be able to visualize the gene expression of this gene. And so the way that's visualized is by a dot plot, where the size of the circle lets you know how many cells of that given cell type is expressing that gene, and the intensity of the color is telling you to what extent that gene is being expressed in that cell type. And we have a little legend over here that explains what I just said with little icons. And you can see here that, as we had seen in our previous example, this gene marks specifically secreting cells and also goblet cells. And we can see that here we have a very strong dot that's marking that cell. Again, this is looking at all the data that we have for the lung, whereas before we were looking at one individual data set. So this tool is extremely powerful in terms of wanting to do quick explorations for gene expression of any gene of interest. I'm going to add another one that I know looks pretty good. And if you hover over any of the dots, it'll give you a bit more information as to what that gene is, the actual values that were used to create the size of the circle and the intensity of the circle, the cell type, and the information of the gene symbol as well. So this is another example of another gene that I know marks ciliated cells. Where are the ciliated cells? There you go. So you can see that the intensity of the dot is pretty strong, and that's because this gene marks ciliated cells. Just to give you an example of what that looks like when we have a lot of genes, I have a very long list of genes here. Give me 1 second. We can copy and paste comma-separated genes. And it'll take a little longer to load, but it'll load. While that loads, I want to talk about what's happening here on the left side bar. Here, there are filters. So if we are interested in specific subsets of the data, we can filter by that. For instance, we can filter by individual data sets, and that'll show us only the data from those data sets. But more importantly, we can filter by disease, ethnicity, and sex. Okay, it loaded. So now here you can see all the distribution of gene expression of all these genes across the cell types. And I'm going to close this section by showing you that once you have loaded your genes, you can hierarchically cluster them, both the genes and the cell types, so that genes that are similar in gene expression will get close to each other, and cell types that are similar in gene expression will get close to each other. We do that by the sorting option here. We can go into hierarchical for cell types. Now all the cell types that are expressing similarly the genes that are shown here are going to be grouped together, and we can also group by genes. And now we can see nice groups of gene expression and start making sense of this data and understand how cell types relate to each other and how genes relate to each other for any set of genes that you're interested in. With that, I'm going to start. I'm going to pass it to Max to talk to you more about the other piece of software. Max, do you want me to drive the slides, or you want to share the slides yourself?
I can share my screen. Thanks, Pablo.
Yeah.
And I hope my Wi-Fi is also okay right now. I'm also in a on the road, so just feel free to ping me if I start dropping out at any point during this. But yeah, awesome stuff that Pablo showed. And so I'll go ahead and talk about another product that we have in the Cell by Gene suite. So everything that Pablo showed you just now was all hosted. So that is, you can essentially go to our portal and access all of those tools. If you do have a use case, where you need to actually work with data locally, so perhaps it's new data that you've generated and you don't want to upload it to any external portals yet, you can use our Cell by Gene Annotate software. So this is a local solution that allows you to work with data. So like I said, new data that you've generated and want to keep it local, but you can also download data sets off the Cell by Gene data portal and work with them in a local setting on your desktop or laptop. And the other cool thing about Cell by Annotate, let me see, I forgot to put it in the slideshow mode. The other cool thing about Cell by Gene Annotate is there is an extra feature here. And particularly what this feature enables is collaborative data analysis. And let me see if I have on the next slide. Yeah, well, so basically what this allows you to do is actually re-annotate data sets. So if you're kind of in the process of analyzing your new single cell data set or re-analyzing someone else's data set, and you start to see markers that subdivide cell types that weren't previously annotated, you can create these custom annotations. You can also share those annotations with your collaborators. So if you're working in a group, either with another tissue expert or with a computational partner, you can share these analyses back and forth. And really this helps speed up the workflow of annotating single cell data sets. So this does require a small bit of coding or scripting, I should say. So in order to install this tool, you will need to install via pip, via the command line. The good news is that's pretty easy. So you can just run pip install CELLxGENE. You may want to do that, and we recommend doing that in a virtual environment. If you don't know what that is, like, feel free to contact me and I can walk you through it. But essentially, it's kind of just creating a sandbox in your computer where this program will be installed along with all its dependencies, and it will be separate from everything else. So there are essentially no conflicts or no issues with trying to update other packages. Once you've done that, you can essentially just type in CELLxGENE launch and then the path to your data set. You don't even need this open flag, to be honest, but optionally you can include it. And then what you'll get some lines printed out to your terminal, and then at some point it'll say launching CELLxGENE, or sorry, launching. Please go to this URL. So you go ahead and paste that into your browser, and from that I'll actually switch my screen here, and I'll also attempt to do a live demo. And so here I've already gone to localhost 5005. And we can see that we've got an instance of CELLxGENE annotate up and running. And so you have a lot of the other functionality, or you have all the functionality essentially that Pablo had demonstrated prior to this. So we can color by categorical metadata, we can color by numeric metadata, we can perform selections of numeric metadata. And Daniel asked in the chat, is it the same for Seurat object? So no, CELLxGENE annotate will not take a Seurat object as input. You will need to download the ANN data H5AD. And if you already have a Seurat object for an experiment that you're working on, there are solutions that you can use to convert that Seurat object to an ANN data object. And if Pablo, you could link SC Easy in the chat. Thank you very much, I'd appreciate it. Good question, Daniel. And so, yeah, all the same functionality is there. What I'll talk through briefly right now is the ability to annotate these new cells, or sorry, create new annotations based on our findings from the data set. So I'm just going to go ahead and quickly color by cell type. And in particular, I want to pay attention to this cluster right here. So all of these cells have been called as fibroblasts. But it's kind of curious because there actually appear to be like two different clusters that are kind of showing up in the UMAP. So we can go ahead and use the differential expression feature to actually go ahead and see if there are maybe any differences between those two clusters. So I'm going to go ahead, as Pablo showed you earlier, and select my first population of cells using this lasso selection tool. And I'll go ahead and assign that to group 1. And then I'll do the second population of cells and assign that to group 2. And I'll go ahead and run that. So we're going to run this differential expression on around 6,000 cells. And that finished more quickly than I thought it was going to. So that's good. And let's see. All right, so we do have a lot of differentially expressed cells. And let's see. Yeah, so basically what I'm looking for here is if there are any meaningful differences in gene expression between these two cells that are both annotated as fibroblasts. And just coloring by the gene set, it looks like there are. I can go ahead and look through this list and see if there are any individual genes. And yeah, there do seem to be some individual genes that are marking this particular population. So we might, in this instance, want to flag this particular set where, kind of separate both of these populations into potentially a new subdivision of these fibroblasts. And so that's what we're going to do right now. So in order to do that, I'll just close this to clear up some room. And let me check. I just saw maybe there's a question in the chat. No, it's cool. I see easy. All right. So if we want to go ahead and create a new category, we can just use the create a new category button. That will bring up this modal, which pops up, and we can create a unique category name. And so what I'll do is I will call this annotation revisions. And I'll just go ahead and create that category. That new category pops up down here. And initially, it will have all our cells data set, all the cells in our data set being unassigned. So if we colored by that, we would just get all the same color there. What we can do now is actually, let me go back to this. And what we can do is now select the population that we're interested in, like kind of subdividing. And we can click on this little plus symbol to add a new label to this category. And so when we do that, we can now say, okay, fibroblasts subclass 1. And if you happen to know like what this group of genes, like maybe they're all you know, implying a similar functionality, you can, you know, you could, the free text field, so you could imply, or so you could say like, oh, these fibroblasts are cycling, or, you know, something more biologically relevant. For now, since I don't know what this list of gene means without some further exploration, I'll just make it kind of general. But with that, you know, you add this new unique label. You can add label and assign to these cells that we've manually selected, and go ahead and do that. And so now we've got basically this custom category that we built, and we can do that, you know, to our heart's content. So we'll go ahead and do that for the second one. Instead of doing the manual selection this time, I'll go ahead and actually select based on the histogram here. So here I'm selecting on those cells that have a low score for this particular set, so that this the complementary subcells to what I had selected previously, although it looks like there may be some overlap. Let's see what happens. I'm going to go ahead and make that, create a new label for the category. I'll call it fibroblast subclass 2, and let's see. And so here, sorry, I did one thing wrong. I needed to unselect this. And now I think, actually this might be great. I'll unselect, actually I realized that that's not going to work because all of these cells will score lowly for the gene sets. Perhaps if I do this, I can select all of the fibroblasts here, select this lower score for the gene sets. And now I think I'm on my way to getting this other class of fibroblasts. And let's see. Okay, so close. This is definitely close to what we're looking at. We could probably also do manual selection for both of those. But yeah, in short, this is how you go ahead and make custom revisions of your annotations or add custom annotations to your data set. With that, I'll switch back over to the presentation and just talk a little bit about some additional functionality that we have started to look at, or that we've started to do work on to aid in the annotation workflow in the CELLxGENE Annotate product. And essentially the concept is this. You guys may have heard of like these new reference atlases that are coming online. So if you've heard of Tabula Sapiens or the Human Lung Cell Atlas, which Pablo just demoed, these are high quality atlases, which are composed of data sets generated typically across many labs, across many technologies, and across many samples. And you know, the groups that generate these atlases spend a lot of time curating the cell types and essentially annotating these atlases, and then also defining the markers that, or listing out the markers that define these cell types that they annotated. And one of the tools that we're building allows you to be able to take advantage of these annotated references and be able to transfer that knowledge that's encoded in here, AKA the annotations, to new data sets that have not been annotated yet. So, you know, if we have an annotated reference that essentially has all of these cell types already defined for each of these cells in the data set, we can transfer that information over to an unannotated data set and do that using the CELLxGENE Annotate tool. And so then we'll have annotations on our new data set. Beyond getting new cell type labels for our data set, we can also get confidence scores for those labels. So if we've marked something as a fibroblast, we can see how confident was the tool in assigning that label to that particular cell. And we can start to tease apart the high confidence predictions for an annotation versus the low confidence predictions for an annotation, and start to look at why there might be low confidence predictions. predictions, because they could either be noise, they could be biologically interesting. It's kind of data or situation specific. At any rate, that's coming soon. We've got a prototype that we're releasing a little bit later, probably Q1. But if you do want to help us test the alpha release of this particular tool, you can reach out to me at mlombardo@chanzuckerberg.com for access to this prototype and essentially to help us test it out. And I'm happy to give you more information if anybody's interested in trying out the tool. And I think with that, we will kick it over for questions or any feedback. And thank you all for the time and giving us the space to talk here to you today.
Yeah, thank you so much. I think we might have been over a few minutes over time, but I don't know if there's time for questions or if there's any questions. But thank you all. We'll drop this presentation and all of our contact information in the Slack channel, so that you guys have access to all the stuff that we just presented today.
And there definitely is time for questions. So if anyone has any, feel free to drop them in the chat, or you can unmute yourself and ask the question.
While we're waiting for questions, Max, do you mind going to the slide where we have the numbers of CELLxGENE Discover, just so that people can digest them? I didn't cover that just for the sake of time.
Yeah.
Hello. Please, I have a question.
Hello.
I'm not sure if you said it and I missed it, but I was wondering, these databases and the gene expression data, is it representative of the human population?
Currently, you mean the data that we host in the portal, right?
Yes.
And is it represented in terms of ancestry? I'm guessing that's the question.
Yes.
Yeah, currently we mostly have data from European ancestry. There's a few from a Chinese population and a few from African Americans, but it's less than, I'd say less than 10% of our full data. We are putting efforts both in the grant side and in the data ingestion to try to bring in more data. But as you may all know, this is a problem of not only obviously a piece of software, but in the entire field as a whole. And we're trying to fill the gaps where we can. So there's not that much. We're trying our best to bring in more of that. If there's some more questions, we're always happy to chat with anyone. Feel free to reach out to us through the Slack or through the emails, and we try to be as responsive as possible. And we're also always happy to come give an extended workshop for specific group of people. If you feel like your lab or people in your lab are interested in learning more about these tools, you can also contact us about that.
Great, thank you so much.
Thank you.